Hello, everyone. Good morning, good afternoon, good evening. Welcome to our third and final session of the webinar for new students on BioCyc. And today we will be hearing about omics analysis tools from Suzanne Paley, who is, among other things, the person who did most of the implementation for these tools. And so she's the expert. And so I will turn it over to her and I will take your questions and comments in chat or QA. Okay, go ahead.
All right, thanks, Peter. So BioCyc has a large suite of omics analysis tools. Today I'm going to be covering some of them in, well, I'll go briefly over most of them in slides. I'll give demos of a few of them, selected ones. So I'm going to be switching back and forth here between slide and demo mode. We have a wide variety of tools for both transcriptomics and metabolomics data analysis, and even multi-omics data analysis. So this is just a brief summary of the kinds of things we have. We have pathway-based visualization of both gene expression, so transcriptomics, proteomics, metabolomics data in a variety of ways. You can even either overlay this omics data on the zoomable metabolic map, the cellular overview diagram that hopefully Peter Karp showed you in the first session. You can overlay such data on individual pathway diagrams or on groups of selected pathways, either pathways you have individually selected because they're of interest to you, or pathways that have been identified by the software as most highly perturbed. And there are different ways of doing that. And I'm going to go over some of those. We have a completely different visualization that we call the Omics Dashboard that creates a graph-based visualization, as it says, using hierarchical systems, cellular function. I'm going to go over that in more detail. We have different ways of performing enrichment analyses. So you can find out which pathways are enriched for a particular set of genes or metabolites, and Go terms that are enriched for a particular set of genes, not metabolites, unfortunately, or transcriptional regulators for organisms that have transcriptional regulation data. We have ways of searching for results of mass spec data, such as monoisotopic mass search. We have a metabolite translation service so that if you come in with maybe a KEG ID or maybe a chemical formula or an INCHI or a monoisotopic mass or something, you can translate that and get the BioCyc ID or the ID in PubChem or whatever that we might have. So just helps translate one ID from one source to another. And we have ways to display data on our Genome Browser tracks. I don't know if I'm not actually going to demonstrate that today, but hopefully it was mentioned during talks about the Genome Browser. So for example, if you have chip data, you can display it on our Genome Browser. So for most of these, most of the tools I'm going to describe today, they all take more or less the same input data format. We're looking at data as tab delimited text. So if you have, say, an Excel file, you need to save it out as tab delimited text rather than Excel format, where the first column is going to contain the identifiers. And what do we mean by identifiers? Basically, anything that our software is capable of mapping to an object in our database. So the gene accession, that's an easy mapping. Common names are synonyms. If we have them in our database, then if those are in that first line, we can map them. So for example, TRPA is a gene name, whereas, well, I can't rattle off a locus tag off the top of my head, but that's going to be a unique identifier, whereas a gene name is a more common name. If you have links to other databases, for example, a Uniprot ID for proteins, a KEG ID or a PubChem ID for metabolites, For some of those, you need to actually prefix them by or if you prefix them by the database ID, then we'll definitely recognize that's what they are. If you don't, for example, a Uniprot ID or a KEGG ID, we can sort of recognize by format that they're probably from one of those databases, so you don't actually need that. If you have, there's some data, I think PubChem has IDs that are purely numeric. So for those, you would actually have to prefix it by the database ID. Obviously, we can use our internal BioCyc identifiers, although you might not know all those. For metabolites, you can use INCHIs or chemical formula or monoisotopic molecular weight. And then if we can also, if you can provide a combination of pretty much everything you know about the metabolite identifier that we might be able to match, and then separate them by a $sign. So in this case, for a particular compound, well, in this particular compound, we have all these links, but if you have a compound name that might not be recognized, you might also provide the KEGG ID in the hopes that we'll recognize either the name or the KEGG ID, because we obviously don't have a complete mapping between BioCyc and KEGG, or perhaps you might also supply the PubChem ID, and hopefully, if we don't have matches to all of those, we might have matches to one of them and be able to identify the metabolite that way. So that's the first column. That helps us map the data values in your file to objects in our databases. And then after the first column, you can have one or more numeric data columns. And there can be other kinds of columns too that we just ignore because you're telling us which columns we need to look at. And these data columns can be, I mean, they're just numbers to us, but we need to know what kinds of numbers they are. So there are really two types of data values that we see. One is like absolute values. So things like counts or intensities, where they start, the lowest possible value is 0 or sometimes just slightly higher than 0. And they only go up in a positive direction. And then we have relative values, which are ratios or log ratios. So there are two kinds of ratios. If you take the simple ratio of two values, this is one experiment versus control, or one experimental condition versus another experimental condition. If you just take the plain ratio, what you're going to end up is a one-centered value. So where values go up, they're going to be higher than 1, and where they go down, they're going to be less than 1, but they're all going to be greater than 0. So that's what we consider a one-centered scale. But more commonly, you see data values where they're log ratios. So there's a 0-centered scale where, if the expression is going up, you have a positive number, and if expression is going down, you have a negative number. So those are the kinds of data we need to know if your data, which of those how to interpret the data is which that kind is. So that's pretty much all we need. And that is the input to our Omics Dashboard, which I'm going to cover next, and also to our all our tools where we display omics data on the on various pathway-based visualizations, the metabolic map, the individual pathway diagrams, etc. So now I'm going to move on to the first tool that I'm going to talk about, which is the Omics Dashboard. So this is an interactive tool for analysis of omics data. It's a web-based tool. It's most useful for gene expression and proteomics data. It can also be used for metabolomics or reaction flux data. And the idea here is you come in at a high level, you want to understand your omics data set at a high level where you have an overview of many different cellular systems, and then you can drill down on particular things that are of interest in an interactive way. And hopefully, visually detect patterns that, maybe might not jump out at you when you do a simple statistical filtering. So we divide cellular function into a set of systems and subsystems. So a hierarchical view. These systems come from multiple sources. Largely, they come from the Metabolomics pathway ontology. So we divide into a bunch of different classes and individual pathways. For example, we have a high-level system of biosynthesis, which has subsystems of amino acid biosynthesis and lipid biosynthesis and that kind of thing. We have a hand-curated subset of Go terms, mostly biological process terms, although we also look at like the outer membrane stuff. So things like, response to stimulus, DNA processing, that kind of thing. Things that are mostly not metabolic pathway based. And this only really works for databases that have been annotated with Go terms, which will not happen. Well, it has not been done for all our databases. It has been done for some of them. And then we have a handful of other subsystems that we can compute functionally based on what they do. For example, transporters, sigma factors, regulons, if for databases that have transcriptional regulatory information. So the dashboard is organism specific. So the set of systems and subsystems is going to depend on the particular organism and the different functions that are in that organism. It's also data set specific. So if you have a data set that, an organism specific, for example, if we're looking at at human, for example, you might find a subsystem for hormone biosynthesis. But if you're looking at a prokaryote, that's not relevant to prokaryotes, so you won't see that that subsystem on your dashboard. It's data set specific. So if your data set only, for example, if you filtered your data set so it only contains significant genes, which is one recommended way of interacting with the dashboard, and your set of significant genes doesn't include any genes in cofactor biosynthesis, then that subsystem will not be included in the dashboard. It's also customizable, so you can add or remove plots, or you can create new subsystems from a gene. For example, if you have a gene set that is interesting to you for some reason, you can create your own subsystems that way. Or if you have a Go term that was not one of our hand curated Go term, but it's one that you're interested in, you can create that way. So the way to get to the dashboard is here under the analysis, under tools, analysis, Omics Dashboard. And I should mention this page here. This is a help page that essentially describes all our different omics analysis tools with links to them. So if you want to be able to access them and forget how, you can look at that. But the Omics Dashboard, this is the input page to the Omics Dashboard. It gives you ways to upload your data. It also provides some examples. We're going to be looking at one of the examples in a minute. There are also other ways to get while interacting with the other omics analysis tools. You can also generate the dashboard. So we're going to be looking at a sample data set that's from E. coli. It's a 10-minute time course following the shift from anaerobic to aerobic growth. And the data is normalized average RNA-seq read counts. So this is the basic organization of the Omics Dashboard. You have a series of panels. So this is one panel for different omics cellular systems. So we have a biosynthesis panel, degradation panel, central dogmas panel, which is transcription, translation, and protein processing. And then each panel contains a set of plots or subsystems. For example, I don't know if you can read this, but this contains amino acid biosynthesis, nucleotide biosynthesis, etc. So what are these? This is a fairly rich visualization. There's a lot of information collapsed here. So I'm going to go over what we're seeing here. So there's, for each plot, we have a set of colored bars with a key up here. So the colors correspond to the individual time points. So this was a time series here. We have time 0 up to 10 minutes here. So each of these plots, these are the time points. On each one of these bars, there are these small dots. These correspond to individual gene expression values. So these will be the individual genes that are involved in fermentation. So you can see all of them at once on this line. And then there's a large dot at the bottom here, or may not be on the bottom, but in the middle here for each of these lines, which indicates the average over all of the genes in the subsystem. So you can see all at once, both the range and the distribution of the genes in that subsystem and the average, and how it's changing over the course of this experiment. So, for glycolysis, you can see it's sort of hanging tight for the first couple minutes, and then it's decreasing. And that's the average, and the range is also more or less doing the same thing. But if things are bunched in a certain way, you might, or if the average is thrown off by a single gene, you can get an idea of that. So there's a lot of information packed in here. What you can't do here is see what the values of the individual genes are. You can just see their distribution. There's also an option to show the sums. For example, if you wanted to, these are RNA seq counts, you wanted to see the what fraction of the cell's energy goes into, or cell's resources go into a particular functionality, you can show the sums for all the genes. And that looks like this, where what we were looking at before is still there, but it's sort of compressed now because the bars show the sums. So that's an option here, and now the sums is checked. So those are the three different types of data that are included here in this particular visualization for each of these systems at a very high level. So the bar height corresponds to the sum of the expression values. So this is our front page. This is the overall start page for the dashboard for this particular data set in E. coli. So this is where you start. Suppose I look at this system and I say, okay, well, there are a lot of genes involved in amino acid biosynthesis. So, overall, perhaps, this is very interesting, but I want to see more. So you can click on a particular plot and you get a blown up view, like a detailed view of amino acid biosynthesis. And so this is hierarchical. So now we're seeing pretty much the same thing, except our panel is amino acid biosynthesis, and the individual plots in here are the synthesis of each of the individual amino acids. And some of them have large numbers of genes involved. I think glycine biosynthesis looks like it only has one gene involved. But again, you can see the same kind of information, the distribution of the data, the averages, and see how they might be changing. And then I can go further. For example, arginine biosynthesis looks like it's pretty steadily going up and then coming down, and I want to have a look at that in more detail. So I click on that, and now I'm finally at the lowest level, where I'm looking at the level of the individual genes and seeing how they change. And different parts of different systems have different numbers of levels in the hierarchy. Some of them are only, there's only one level, and some of them there's more than two. This is a case with two levels, so it's nice for demo. But I can see, pretty much all of these genes are going up and then coming back down at the end. So that's useful. They're all behaving more or less the same way. From here, there's a button to show the pathways, and that will pop up a diagram of the pathway with the omics data superimposed on it, so I can see how it's a different color scheme, of course, because the colors are by value rather than by time point. But so I can see how it's what this looks like in the context of the pathway diagram. There's also an option to show operons, which shows how the genes in the pathway are organized into operons. This does not actually have the omics data superimposed on it, but at least I can see how the genes are organized into operons. Under the options menu, actually, was there anything else? No. Under the options menu, there are other options. For example, I can export the image, I can show a table, I can do various other things. But for databases that have transcriptional regulation data, such as BioCyc and a few of our others, you can also show a view of the transcriptional regulators for this system. And what that does, so now since this is BioCyc, these genes are controlled by this set of regulators. And so now I can see a graph of how they're changing over the course of the experiment. And then I can filter that by say, okay, well, I really just want to see FIS and what that does. So if I click on this here, it will show me just the view of which of the genes are regulated by that transcriptional regulator. It seems to affect both some of the other transcriptional regulators and these genes. And so I can see just how those are affected. So this is a very powerful visualization. It does a lot of, if we have all the transcriptional regulation data, it packs a lot into a small amount of space here. So hopefully, and these visualizations are only available at the lowest level where we're looking at the individual genes. But other things we can do, you can choose whether you want to show log or linear scale. That's only the case if your data is not already log data. You can customize it in various ways, the panel size, the vertical scale. You can sort your genes in various ways. You can interactively, if you have multiple time points, you can interactively select or disable them. You can average the data for multiple replicates if you have that. You can actually perform an enrichment analysis. You can generate a table of data, and you can export the data, the whole visualization. You can export it to a smart table, which is just a table of the data, but you get a link to quickly, from a smart table, you can quickly regenerate the dashboard display. So that makes it quite convenient. So in general, when we're using the dashboard, ideally, you want to apply your normalization and significance calculations before uploading the data, because that is going to get rid of a lot of noise, because sometimes things don't look like they change very much, and you end up with, and it's because there are so many genes involved that are not doing anything, that they wash out the genes that are, the averages don't seem to be changing very much. You have to decide, if you have multiple replicates, you have to decide whether you want to average them before the upload or have the dashboard do it for you. If you have, the dashboard can do it for you, but it's much slower when it does that. I mentioned the data format of the data that you have to input. In addition to the types of data values, for example, we can have fold change values or absolute quantities. Your data values can also include p-values from replicate analysis. And you're not going to want to display those in the same way that we display the other data. But since we can filter in which columns we're going to show, you can not show those, and then you just use them for the enrichment analysis. So the enrichment analysis, which I'll talk about briefly, we have a number of different ways to do enrichment analysis. Most of them are via the smart tables, but this one is via the dashboard, because we want to see which systems have more perturbed genes than you would expect by chance. So in this case, for each data column, you need some kind of measure of whether or not it's significant. So that could be via a p-value, a separate p-value column, or you can maybe specify a threshold. For example, all genes that have, a log fold change of more than 2 or something like that. And we apply a correction function, and we compute an enrichment score for every system and subsystem. And then the panels show both the enrichment scores for each system and then the highest component subsystem score if it's greater than the enrichment score. So here's an example. So what we're seeing now, we're seeing the same set of time points. The bars represent the enrichment score for the for each system. So, and that's what the numbers here show. But then in some cases, you have the overall enrichment score might be low, but there's quite a high enrichment score for some subsystem under there. So maybe amino acid biosynthesis isn't that enriched in general, but clearly there's something going on here, because there's at least one subsystem under amino acid biosynthesis that is highly enriched, but it's just the overall enrichment score is lower because it's being swamped out by all the other subsystems. So the example the slide looked at was the protein metabolism. Again, these are very low scores initially, but clearly there's something going on with some subsystem. So I looked at that, and I can see although there are lots of genes here in other systems where there's not much going on, all the action here is in the protein maturation. So that helps me drill down there. So let me just give a quick demo. I'm actually not sure that I have time for this, but I have preloaded the Omics Dashboard here on a screen. This is our example data set. So I just want to show, for example, here's carbohydrate biosynthesis. Let me click on there. That's just a single level. I have the individual genes. I can show the pathways. It looks like it's only one pathway for this organism. So this is pretty much, actually, this is pretty much what I showed you before. But we also have things like the central dogma, for example. I can click on here. Somewhere I have multiple hierarchical levels. DNA metabolism, perhaps. Yeah, so DNA metabolism can be, replication, repair, etc. And then I can drill down further to see what genes are involved. Etc. There's also some number of genes that are not present in any subsystem, and I can show those if this is just the gene, so there's no hierarchy here, but I can see what they are and if they're doing anything interesting. In my options menus, I can customize the panel in various ways by editing the components, changing its name, etc. Sorting it. I can also add new, I can edit my display preferences, turn on or off different time points, add a new top level panel. And if I create customizations, I can then, they're not saved on BioCyc, but you can download your set of customizations and then upload them back up again if you wish. So this is a very powerful tool. It does a lot of different things. I can change the size here, the font size. Whoops, that's not what I meant to do. There, I changed the size of it, and I can also increase the font. And I can export this to an image if I want to use it for publication, show the data just for this panel, etc. So there's lots of things I can do here. But I think I should move on, because the other major thing I wanted to talk about is showing data on the cellular overview. So you're hopefully familiar with the cellular overview diagram that I think Peter Karp showed in the first tutorial. You can overlay omics data on this. So this is transcriptomics data overlaid, so each of these reactions are going to be catalyzed by an individual gene, or one or more genes. And so we color code the reaction lines based on the transcriptomics value of the gene involved. And then we can show these little pop-up graphs to show how that, we can run it as an animation, as a time series animation, or we can, for individual genes or individual steps, we can pop up these graphs. And I will show you those in more details in a minute. So we can do that for transcriptomics data. For metabolomics data, we can do the same thing, except now what is colored is the metabolites instead of the reaction arrows. Actually, let me, this is showing zooming in, but actually let me do this in demo because that's going to be more interesting. There we go. So I have, well, let me actually show you how I got here. From the tools menu there, I went to the cellular overview, and here's my cellular overview. And here in the right operations menu, in my tools menu, there's an option to upload the data from a file. I can also, there's also options for entering data in other ways. GEO is the Gene Expression Omnibus database. And so I can upload data directly from there if it's one of their data sets in the particular format. but the most common way of interacting with this is uploading data from a file. So this is, it shows the example of the file input. I need to specify the file. I need to say what, I need to provide the information that we need to know to interpret the data. So for example, the files, the items in the first column, in this case, they're genes. They could also be metabolites or proteins or reaction identifiers. I need to say which of the columns are my data columns, and it could be just one or it could be multiple columns. And then I need to say whether it's relative or absolute, and if it's relative, whether it uses a 0-centered or a 1-centered scale. And there are help icons here to tell you what these mean. And then I can actually bring up multiple omics viewers at once. So this shows how they interact a little bit. So by default, we're going to be showing data on this diagram, but I can, in addition or instead, I can also bring up data as an option on the Omics Dashboard, as I've just shown you, in a new tab, or as a table of pathway diagrams, or as a pathway collage. And I'm going to cover pathway collages a bit in a little bit more detail in a minute, but these are some of the options that we have. I happen to have a data set preloaded here. So what this data set is, this is a reaction, or this is response of E. coli to exposure to peroxide. It's a time course. So initially this comes up, I have this control panel. I can't actually see very much here. So the first thing that I think I want to do is I want to increase the width of the highlighted edges so I can see the colors a little better. So I'm going to do that. And so what I can do here, let me drag this a little bit. So I'm showing, this is the 5th column of my data set. I'm showing columns 5 through 9 here. So it's showing that the first one that I uploaded, which is column 5. I can play this as an animation and I can see things change. This is a histogram of the data and this explains my color key. So, most of the data falls in the central region here, but it's really the outliers that I'm likely to be interested in. But if I play this, I can sort of see how things change here. But it's kind of hard to see, that's a lot to visually keep track of. So I can actually change things in various ways. For example, I can customize my color scheme. So I prefer to see fewer colors here. So I think I will switch to this one, which gives me fewer colors. And then perhaps I want to see red for a lot. This color scheme is customizable. So actually, let me make this symmetrical drag. So if I drag this down, it's going to drag on both ends. So now more stuff is going to be coming in red and purple. And then what I really don't want to see is all the stuff in the middle that is cluttering everything up. So I can click on this and just turn those off. So these I can also type in if there's a particular value that I want. And so now I see very clearly the stuff that is popping out as being up or down regulated. And now when I play my animation, I can see stuff turning on or off over the course of it. So I can very clearly see what is changing here. So let me stop here and say, so let me mouse over one of these. And so this is giving me information about what reaction I'm looking at, what its genes are, et cetera. But there's also a little omics button here. And if I do that, I get the pop-up so I can actually see what this particular gene is doing over the course of the experiment. And I can do that for any of this. So this step happens to have 2 genes. And so these are draggable, so I can look at them. So they're not covering things up that I care about. So I can see how an individual reaction step of interest, what its genes are doing over the course of the experiment. There are actually preferences here, so I can actually control what these pop-ups look like. The default is this bar chart, but But I can also change it to a plot. Let's just do the most recent pop-up. So that will change it to a plot, and or I can change it to a heat map, which is more useful if there are a lot of genes involved in a particular step. So those are my options here, and I can of course close it if I wish. So this is a completely different way of looking at our omics data in the context of the pathways for, well, the context of the pathways for the entire organism. I think that's, I can also output this to the dashboard or to the, to a smart table so I can have access to this data again. Let me move back to my slides. I can also put gene expression, actually, let me show that again. I can put gene expression data on an individual pathway or metabolomics data on an individual pathway. Let me get, so if I click on one of these pathways, for example, this is a link to the pathway. If I come here from the cellular overview diagram, it's going to automatically have the omics pop-ups. If it doesn't, because I have not yet loaded omics data, then I can actually, if I were just to go to another pathway, for example, well, do I have a link here? I do not have a link here. Here's some other pathway. Oh, okay. So because I have an omics data set loaded, it will automatically show it. If it didn't, there's an option to customize the pathway, and I can say, show omics data on this pathway, either use the most recently loaded data set, which it has, or I can upload data from a new file. So this is available. So currently it's showing the omics data because I have my omics data loaded. And I've come from the cellular overview, but if I had not and I wanted to add omics data, I could do that. And the same with metabolomics data, as in this example here, where it would be attached to the individual metabolites rather than to the reaction steps. I can also generate a table of most perturbed pathways, and we have a statistical formula. It's a very simple formula, actually, for deriving what we call a pathway perturbation score. This doesn't work for absolute data, such as RNA counts or whatever. It works for relative data counts, where you're looking to see how much things have changed. So we identify what things are, which pathways have changed most. We have a score for them, and we can generate a table. So this score essentially measures the extent to which a pathway is up or down regulated. I'm not going to go over the math of how we calculate them because we're running out of time here. But we do computer score, and then we display the most highest score. You select, do you want the 10 most highest scoring pathways, the 20, whatever, and then we can display those as a table. We can also generate pathway collages. I'm gonna get to pathway collages again in a minute. Pathway collage is a set of pathways that you pre-select that we build into a single diagram, and it's customizable in various ways and editable. We can also do an enrichment analysis with smart tables. So the idea here is that we have some experiment. It generates some set of genes or metabolites that are highly perturbed or highly expressed or whatever, and we want to know, what does that mean in the context of the global action of the organism? And so given a group of genes, we might want to ask what GO terms are statistically overrepresented in that set, or which metabolic pathways are overrepresented, or for databases where we have transcriptional data, what transcriptional regulators are overrepresented. If we were using metabolomics data, then we can ask which metabolic pathways are overrepresented. So we start with a smart table of in this case metabolites that just the set of metabolites in our data set, and then we can run a run the enrichment analysis, and we get a set of pathways sorted by p-value. So in this case, I don't actually know what this data set is, but in this case, clearly the NAD biosynthesis and tryptophan utilization seem to be the most highly enriched sets of pathways. We can also do a pathway covering analysis, where if we have a set of input metabolites, we want to find out which set of pathways in that organism account for all the different metabolites. So I'm not going to go over this in any detail, but essentially you supply a file of metabolites, and in this case, we will have identified a set of pathways that cover those metabolites with links to the pathways, and so these glyphs have the metabolites highlighted that are relevant here, and then with links to the individual compounds and to the pathways. I mentioned briefly that we on our Genome Browser, we can show chip data or other genome tracks. So hopefully you've seen the Genome Browser. I think one of the Peters would have showed it to you in the previous tutorials. You can set it in a mode where you show tracks, and then you upload your data, and you can see it as a graph here. I don't actually have an example to show you, but here's an example slide. So let me move on now and talk about pathway collages in a little bit more detail, because that's the other tool that I was going to demo and show in more detail. So a pathway collage is an application where you specify a set of pathways that are of interest to you for some reason. And they could have been pathways that were identified in the course, by the pathway perturbation scores that I mentioned before, or they could be ones that you have pre-selected because they are of particular interest to you. And it's a diagram that combines those pathway diagrams in a way that is zoomable, editable, customizable, helps you generate high quality images for publications and presentations, and you can overlay omics data on it. So this is an example. We actually have, I don't know if you can see the light gray backgrounds, but we actually have 1, 2, 3, 4 pathways here that are that are combined with connections between them, and we're actually showing in this case reaction flux data on these. No, actually, this is looks like transcriptomics data on this pathway, on this pathway collage. And so you can edit in various ways. You can adjust the positions of actually anything you want. You can edit the labels. You can customize the appearance as far as color and thickness, font size, shape in some ways. You can show connections between pathways. You can merge duplicate sections of the pathway or delete parts of it. You can add additional pathways that involve a particular metabolite. You can highlight different things of interest. You can show or hide omics data. And these are all live diagrams, so they have links to the BioCyc pages. And you can save or reload or export to PNG. So this is, again, the old menus. So let me actually, I will show this in a minute, but actually, let me show this right now. So from the tools menu, I can go to pathway collages, which will give me information about, examples and et cetera. If I've previously saved a pathway collage, I can just go to the pathway collage viewer without anything loaded and load from a file. But for generating a new one, it gives me options. I can either create a smart table and then export it to a pathway collage. I can do it from an individual pathway page. I can do it from the cellular overview, or I can this just gives me a list of all the pathways in my organism, and I can select which ones I want. That can be somewhat tedious. So what we actually recommend is generating a smart table that contains the pathways that you're interested in. And then you have it, and you can always regenerate the collage. And there's an option here in the export this is the normal sidebar menu. Under export, there's an option to export pathways to pathway collage. And I've actually already done this. This is how it comes out initially. So I can zoom in here, or zoom out. If I zoom out far enough, I lose my labels, which in this case is not useful. But if I had a much larger pathway collage with many more pathways, then that would be easier. I can drag things around if I want. Or I can drag individual nodes around, etc., to customize it that way. I can highlight individual metabolites or reactions, actually, by right-clicking on things. There we go. So if I look at the menu, first of all, I can highlight this, but the presence of this option in the menu indicates that there are places where this is aspartate I'm looking at. There are places where aspartate is present as a side compound for some reaction, and by default we hide all the side compounds. So I can say, well, I want to see all the ones for this particular metabolite, and it's over here. And then I can say, show connections for this metabolite, and it will link it up. So maybe I want to rearrange this so things that are connected are closer together. So I can do that. I can also show all connections, which looks a little bit more messy in this case, but it might help me here. If I move this over here, I can see how these things are connected more. Oops. Etc. Pop this up here, and I can move things around this way. So customize it in various ways. This way, I can customize the, or I can highlight something. So for example, let me do this again. Okay, maybe I want everything here to be red. Just the background. No. Set the node color to be red. And maybe increase its font size a little bit. And make it a hexagon. So I've done that. It's a little. And I either have the option to just do it to just this selected node or for everything for this metabolite. So let me do it for all of them. And so you can see now, it's colored them. I can also change the name here, and it will do that. So I can highlight things in various ways. Okay. I can turn off my highlights or turn them on or off as I wish. But the other thing I can do here is I can show my omics data. So it remembers my most previously loaded set of omics data, which, if you recall, was the one I loaded onto the cellular overview diagram, the peroxide data set. And so now I've got, it swapped out the enzyme names for the genes and their omics data values. So I can see this as a diagram. There's also, it's also customizable in various ways. Well, let me show the customization first. So I can say for everything I want, if I want these are the metabolite labels, if I want them to be, let me do that. So now I've changed them all to blue. I can globally change the font size, etc. Specify whether the icons are shaded or hollow, etc. And give them bigger or smaller borders, etc. And there's separate customizations for enzyme and gene labels. The pathways, I can specify whether or not I can show the background boxes. If I turn off the background boxes, then I can do Shift Select to select a set of nodes. And then I can move those as a group. The reason I can't do that with the background boxes is because then it will identify the background boxes as nodes and it will just take the whole pathway. But I can turn on or off the pathway labels. I can change the reaction widths, etc. So customize this in various ways to make it look pretty in ways that I want. Yeah, anyway, you can play with this. I can export this to a PNG or SVG file. I can save it to a file that can be loaded back into this application. This gives you help about some of the other operations. Oh, one thing I should say, I can also merge things. So for example, right here, we have a duplicated reaction here. So I'm actually going to merge this. Actually, I'm not going to do that. I'm going to move things around first. So it doesn't look quite as ugly. Let me show this. Oops. Okay, it's not moving. Not quite sure why that is. But okay, let me do a merge here. It's going to say, it's going to highlight the other places where this compound appears. And if I merge, oops, I canceled. Let me do that. And let me click on the node that I want to merge with. And now it's merged those two. And I actually want to delete this one because it's also a duplicate. Etc. So I can make my diagram look pretty in various ways. So this is a powerful tool both for visualizing selections of omics data on selections of pathways that are of interest to you in particular, but also for generating diagrams that look the way you want for use in your presentations or publications. I think that might be all that I was going to cover today. Let me see if I have any more slides. Nope, that's it. So I'm happy to take any questions.
Suzanne, yeah, we had we've had a number of questions in the Q&A, but the one that I wasn't able to answer was how can we take into account replicates or standard deviations? And I assume that I'm wondering if there's a way you can do that in the dashboard. I don't think there is in the other tools.
So there is not in the other tools. You would have to apply your statistical analysis first and filter them out and do your significance calculations, and you take your averages or whatever. For the, let me go back to the demo. No, let me go. The Omics Dashboard. We actually have an example here. So the example that I showed you, these two examples here are actually the same time series data set. But the first one was fast, and because it had already just filtered out the most significant data sets and pre-averaged the replicates. This more complicated one contains the complete data, and it includes all the replicates. So I did not have this preloaded, so hopefully it won't take too long. But what you can do is you can specify groups of replicates and which time points, which data columns map to which. Here we go. Almost there. Okay, so you can define a series grouping. So for example, you tell it how many groups you want. So in this case, there's some large numbers of, I think it's 6 time points with 3 replicates per time point. So there's 18 data columns here, plus some additional p-value columns. So we decide we want to create 6 groups because there are 6 time points. And then we just have to say which of our initial columns map to which group. And then we do that, they're averaged, and we can name them, this is this is an example that's pre-generated. So this is already done. So now when we drill down, let me show one that has only one level of, oh, come on. It's a bit slower. So now we see the individual genes, but in addition to just the bars for the individual genes, so you've got the average here, which is the big dot, and then we see the range and the other values. So there's 3 values for each of these time points. So you can see what they are. So we're not looking at standard deviations or anything here, but we do get a sense of, the range of variability for these particular replicates. And the mouse overs here, this tells you what the actual averages are and the actual values for the individual replicates. So again, as I said, when you have a lot of data here, and then we're having to do the average, the tool is a bit slower to interact with, but we can handle that. Our other omics analysis tools really don't handle that at all. So you're going to want to average your replicates before you submit the data. I hope that answers the question.
Okay, there's one other question which may actually, I think, be a little bit out of scope, but I'll throw it out anyway. Thanks. Is there a way to map Uniprot E. coli pan-proteome identifiers to EcoSyc identifiers?
So that would be the a metabolite translation, sir. Oh, no, that's metabolites.
Yeah, that's it.
We can generate, you can look up individual Uniprot IDs. I think if you supply Uniprot IDs as the first column of your omics data file, it will map them to proteins if we have those links, which we should for most of them. I'm not quite sure what you, well, I haven't actually tried that. If you create a smart table.
Yeah, I actually suggested that they try typing a couple of those identifiers into a smart table and see if they translate in a protein column.
Yeah, and it might actually. So, well, I don't have a file, so there's nothing I can do to try, but I would try with a smart table. So if you create a new smart table from an uploaded file, so yeah, this uses the same format supposedly as our other stuff. So if the first column of your file contains Uniprot identifiers, then it will try and find the corresponding protein. You tell it to look for proteins, not genes, because the Uniprot identifiers correspond to proteins. And you can do that. That will give you a smart table of our gene or our proteins. It won't actually include the Uniprot identifiers. But if you have a smart table with our proteins, you can map those, you can add a column that contains the Uniprot identifiers. And Peter, I'm assuming you didn't actually show this.
No.
Well, last time when, let me see if I have a smart table with not too many. Well, here we go. This is a set of proteins. Let me just delete some columns here. So we can add a column for database links and say, I want to see Uniprot. And so for all of these proteins that have Uniprot links, I can generate the table that way. So that's going from our objects to Uniprot. Going from Uniprot to our objects, you would just have to upload a file that contained them. And you would get our objects, I believe. So I hope that answers the question.
Yeah, I think it answers the question. I will point out that these are that these Uniprot identifiers are for this particular strain of E. coli in particular. I don't know about the Uniprot pan proteome identifiers, whether those will work or not, but certainly for individual strains, we have.
Oh, no, for pan proteome, it will not. I thought you asked about E. coli.
Oh, no, it's the Uniprot E. coli pan proteome identifiers.
Oh, so no, it will not do that. You have to have selected a, you will have to have selected a an organism first, and it will try and upload those it will try and interpret those Uniprot identifiers in the context of the current organism. So it will only find matches for ones that exist in the current organism. We cannot do the pan proteome ones, unfortunately. So I'm sorry about that. I misunderstood the question.
Okay, well, that is actually it. So.
Okay, well, thank you all very much.
Yep, thank you.